to a different. I recognize a few faces, but I always tell folks as a researcher in the division of nephrology, it's really exciting to be around nephrologists are really smart people. This afternoon, it's really a pleasure for me to be in front of cardiologists. I think they're even smarter. And I can say that because I don't think Alfred or Island are all they're certainly not in the room, hopefully not on the call. Just really a pleasure for me to be here. So I'm going to give you an overview of our research, which is leveraging what I call big data. It's a relative term, and one person's big data, another person's small data, but I'll leave that to you. And unique cohorts that I think for me were really an attractive opportunity to come from Boston to Utah and start our research program. So along the way, I'll tell you about some of how we're applying these or using these resources. Some of the genes that we've pulled out thus far. It's going to be genetic talk. And then also some of the resources that we're building to really expand the work that we've been doing. So I like to show this overview because it kind of frames what we think of in terms of big data usage in my lab. So we have a lot of our work. It certainly starts with a human sample or a human data point, and we want to then investigate that person, whatever phenotypes we're interested in, mostly diabetes and diabetic complications. And our goal is to eventually bring that back to the clinic. That's how path. For us, that starts with identifying people who easily phenotype them that we use electronic medical records and data. And for us, we're really fortunate to come to the University of Utah, where electronic medical records have been around for 10, 15, even closer to 20 years. I'm a little bit embarrassed to say that the institution that I came from, the job of diabetes, has been around for 100 years. Didn't begin their electronic medical records to happen by the year in about 2018, 2019. So we have this really rich data set that if we pull all of that data together, we have this longitudinal retrospective data, prospective data, and deeply phenotype patients for whatever phenotype is interested to us. Another unique aspect about being in Utah, the large families that have access to the Utah population database in the LDS church, the genealogical record that they maintain. So the geneticists, that's really great and powerful. It's like being a kid in a candy store. You take these individual massive pedigrees, very large pedigrees like the one I'm showing you here, and try to identify traits that co-segregate in those families that are enriched for that trait, and then you leverage next generation technology to identify the molecular basis of their disease. So when we do that, we collect samples and codes. We've established some biorepositories. We've given them various names over the years. Our cohort right now is about 2,000 individuals from various collections that we've assembled: blood, serum, plasma, and urine. We have about 250 ppm to establish iPSC lines. We have some kidney tissue, etc. Are all great resources to do the work that I'm showing you in the right, that omics research. And our lab touches all 5 of these spheres. Yeah.
I'm sorry to interrupt. Just a quick question, and I'm assuming you're going to get to this somewhere. So is it possible to have type 1 and type 2 coexist, essentially? And how does that impact the segregation or co-segregation ability?
I don't think so. I mean, there's certainly different manifestations. Right? Type 1 is where you aren't producing endogenous insulin. Type 2 diabetes, you can have beta cell fatigue, where that person got on some other treatment, oral agents, metformin, for example, and then, just because of the difficulty in treating it, they may morph to insulin, but I would still consider those distinct. Look, ideology is distinct, manifestation is distinct. Okay, at least in my mind, but at least that's how I do it. So our lab has all of these spheres. Mostly for today's talk, I'm going to talk about the one in the upper left, genomics, but we are doing various genomic DNA methylation studies. We're doing some transcriptomics, RNA sequencing, some proteomics studies, leveraging our samples as well as the UK Biobank, which some of you may be familiar with. My colleague Will Holland in the room has helped us to do some various lipidomic studies, and we're really excited about that. And I'll be touching on some of that work as I go along. So the very first thing that I did when I arrived in Utah was to really dive into the electronic medical record system, and with the help of Jose, who was a graduate student in the lab at the time, established what we call the Utah Diabetes Database. Essentially what we asked were the folks here at the EDW could they identify all patients with an ICD code of diabetes and give us all the various labs, medications, EMR data that we might be interested in so that we can phenotype those patients. When they did that, we had about 100,000 patients. And we got literally millions of lab data, millions of blood pressures, vital signs, millions of medication data. And Jose was really great in integrating that together. We then took it another step forward and made some friends at Intermountain and asked them if they could do the same thing. And when they did that, we almost quadrupled our samples from 100,000 patients in the University of Utah healthcare system, adding 317,000 patients from Intermountain, aggregating that all together to a database of over 370,000 individuals. Those of you who have your calculators up know that 100,000 plus 317 does not add up to 371. And that's one of the really cool things about what we're able to do. There are patients that had visits and data points in both data sets. So we were able to aggregate that together, come up with this much richer phenotypic database for our research. And Jose did all of the heavy lifting for that. We're really grateful for him and his time that he spent doing that. Yes, Robert?
I can't resist asking. The numbers between Intermountain and you are quite dramatically different. Is that hospital size or are there structural issues locally that we can do to enhance our numbers? Or that Intermountain may see that many patients?
My understanding is the latter. They have a much larger slice of that patient market share, if you will.
Wow.
So the people that I shared these numbers with were much more familiar with the landscape here at the time. That seemed reasonable to them.
It's something.
Yeah, and I assume that's still true again.
Yeah.
A couple years ago, we've done it. One of the things that we've tried to make this database is dynamic. So every 12 months to 18 months, we refresh this database. So we're getting additional follow-up data for the patients in there. We're having new patients come into the database, et cetera.
Well, I think one thing here, Robin, is that it doesn't say IMC there. It says Intermountain Healthcare System, right? So they have all these hospitals throughout Utah. This is not just the valley, right?
Correct.
And
Maybe they have a broader.
I was expecting actually that they would have even more than 1 to 3 because of this system that is so much larger. But it's a good reminder of why every time we figure out a way to make the red work with the blue, it's so much more powerful.
Absolutely.
And when people like Marcus and others, we have done that a lot in our recovery program, happy figure out the way to do it. And CRPI has a good track record of doing that. It always adds a lot of power to the studies and the grants.
And we were very fortunate that Alfred was that medium for us to meet with the then Srinivas, who was the head of nephrology at the time at Intermountain. It's very collaborative, and a lot of folks told this couldn't be done, but we were able to do it. We were really excited about that.
Not to mention that if you put together the grant and you show that you control 15% of the continental area of the country by showing all these patients that they come from the surrounding states, it's hard for this review panel to ignore that statement, especially when they want to say that they will give money to the whole country and not just to the cohorts. So that's another strong argument when we work with them, because we can just together claim 15% of the country.
Absolutely. So I won't go too much into detail to this other resource that we were really excited about. Was a draw for me to again lead Boston from the University of Utah and see how we could integrate this to our research program that we were trying to start. It's the Utah Population Database. This was established by a government order several years ago, actually in 1982, and it has a genealogical record for over 8 million people. And that's just incredible. It goes back some, 10, 12, even 17 generations for some folks. And in some respects, this has been intertwined with electronic medical record data. We haven't leveraged that aspect of it. We've done sort of this integration on our own, but in theory that exists where this is sort of a little bit more user-friendly and folks can reach out to the Utah Population Database to study whichever phenotype they're interested in. So This is a pedigree that some of you may have seen a picture of, but I don't know if anyone knows that individual in the middle of this pedigree. That gentleman is Joseph F. Smith. He's a nephew of the Joseph Smith, and he's flanked by his 6 wives and surrounded by his 46 children. And this is maybe an extreme example, but this is an example that Mark Skolnick recognized in the late 80s, early 90s as being a powerful tool to understand the diseases he was focused on for breast cancer. And he is very well known for leveraging this kind of resource and to identify a locus on chromosome 17q using the state-of-the-art genomic technology at the time, which were linkage studies. Essentially a fairly sparse set of microsatellite satellite markers across the genome, mapping these in pedigrees and doing some linkage analysis. And he was very fortunate using just 18 pedigrees that had anywhere from 4 to maybe 20 cases of breast cancer to identify a locus on chromosome 17q that turned out to be the BRCA1 locus, sperm myriad, made Mark Skolnick a very famous guy. So that's just one example of the power, maybe one of the earliest and most famous examples of the power of these types of large pedigrees. So before I get into our research, I think it's always good to kind of frame what are we talking about in terms of genetics and genetic studies. And we usually think of this as a function of the allele frequency, which is on the x-axis, and the odds ratio, or the effect size that we detect using various approaches. Our lab does a lot of this, so we get common variants that have a minor allele frequency of greater than 5%, using what is called genome minus rotation study. Some folks in the audience might be familiar with that, but I'm not going to talk a whole lot about that today. I may mention it just briefly. Most of our efforts have been focused in these realms, looking at either what we call rare low frequency variants, so 1 to 5% minor allele frequency, or even less than 1%. And for that, we're using next generation sequencing technology, which I will talk about quite a bit. Things like whole genome sequencing, and I'll mention whole exome sequencing so that we're all kind of on the same page. With these technologies, you identify variants that have different effect sizes. UOS are primarily how to identify low effect variants. So odds ratio is about 1.2, 1.5 max. Whereas with these other technologies, we're looking at rare variants to identify that really highly penetrant major effect variants that predispose to disease. I like to show this because to me this is really what's super exciting about the field that I'm involved with. So when I was a younger person and the first genome was published in around 2000, before some of the folks in here were born, that first genome cost about $1 per base sequence, over $3 billion. And we were always promised that, the goal was to get this $1,000 genomes. And they kind of mapped this out using what a computer science term called Moore's Law. And we expected to get there at some point. We've actually exceeded that 1,000 genome. And this was only about a year and a half ago or so when Illumina used their new NodeSeq instrument. So prior to June of 2023, the cost was about $1,300, maybe $1,100 per human genome. Now that costs about $350. So cut in third, which is just incredible for me. So we've always been trying to maximize our sequencing dollars and go lower coverage sequencing to try to take that cost benefit. But now that's kind of out the door. We can see that a person's entire genome, $350. If you are budget strapped, you can do what we call a whole exome sequencing. We're only looking at that 1 to 2% of the genome that encodes for a protein, and that's under $100. We're sort of abandoning this because it's so affordable. No reason to just limit ourselves to that coding region. What we know is the other 98% is also pretty important as well. So what phenotype are we interested in? So Scott also mentioned that when I was in Boston, I was fortunate to train under my long-term mentor, Andrzej Pezzolesi. He's been a Jolin for, almost 40 years now, maybe. And he has really been a proponent of this phenotype called progressive renal decline. Those of you who are clinicians certainly have heard of albuminuria as the clinical marker of diabetic kidney disease. That's where albumin spills into the urine, there's some kidney damage. Andrzej has always been a proponent that no one has ever died from albuminuria. What people are really dying from is reduced renal function, being on dialysis, and all the complications that go along with that. And he showed this in real time that these two clinical phenotypes are really distinct. That working? I think it's battery there. Battery's there. Can folks in the back hear me? My wife tells me I do speak pretty loud. So the lady in the left confirming that you speak loud. Okay. So he showed this in real time that these two clinical manifestations of diabetic kidney disease, while correlated, are somewhat distinct. And that's a picture of these images here. So he followed patients that had proteinuria, type 1 diabetics, for 10 or 15 years in real time during his career, and showed that despite having albuminuria, there are some folks who plot along the Y to the x-axis is time. These data point to the measure of estimated glomerular filtration, how good your kidneys are functioning. Over 10, 15 years, these patients, despite having high levels of protein in the urine, will never progress to end-stage renal disease, which is an eGFR of less than 15. They'll never go on dialysis. But in the middle panel are folks who progress to end-stage renal disease rather quickly, some over a 10-year period, some over a much shorter period. And then there are other folks who have this in the lower panel, have this non-linear trajectory of eGFR decline. So as a geneticist, this really intrigued me. These are the people that we should be studying. These are the people that may perhaps be genetically predisposed to kidney function decline, rapid renal decline. And can we identify families using some of the resources here that fit this phenotype and then try to uncover genes? So very early on, once we made that connection with the University of Utah EMR system, we asked that question: can we identify pedigrees where this phenotype seems to be clustering? And again, at that time, we only had about 100,000 patients. We identified folks who had at least 3 eGFR measurements over 1 year, and we calculated their trajectories of renal function decline. Altogether, we identified more than 2,000 rapid decliners. So not all of these folks had enough data points that we could assess that. I think we had about 15,000 people that met our criteria, 3 measurements over at least 1 year. So we could establish that trajectory. But 2,000 of those folks were rapid decliners. The others were either slow decliners or moderate decliners. We then took those 2,000 rapid decliners and mapped them, asked the question, could we map them to pedigrees in the population data? We could. Using some of their statistics, we were able to identify 51 high-risk pedigrees. Shown here are just some of the representative trajectory, very similar to the slide I showed in the previous, or the data I showed in the previous slide, but this is the University of Utah data, slow decliners, rapid decliners, and here's the distribution. So we did that for other phenotypes as well that we're interested in, type 1 diabetes, type 2 diabetes, and we can use the EMR data, identify large numbers of individuals, map them to pedigrees, identify what we call high-risk pedigrees, shown in red, using some of the statistics that the Utah population database has. And for the work that we've been doing, we've been primarily focusing on these 460 families. This is Intermountain data and University of Utah data, all linked to the Utah population database. And folks that have rapid renal decline defined by losing at least 5 ml per minute per year.
So I have a question.
Yeah.
So how does this rapid decline phenotype, how does that correlate with the general population beyond just Utah?
So what are we losing per year? Again, it's sort of the physiological rate of renal decline. You call 1 ml per minute per year.
Oh, okay. Sure.
This is an example of one of those pedigrees that we identified looks like. So mapped on this pedigree are about 94 folks that have diabetes, mostly type 2, and that we have established that trajectory of renal function decline. 70 of them are slow decliners. 24 of them meet our definition of rapid decliners. Slow decliners are the open circles or squares. Rapid decliners are those that are shaded. They're color-coded based on whether they are in the Intermountain system in blue, University of Utah system in red, or in both in green. You can see this is a pretty large pedigree, goes back several generations. Really exciting to be able to work with this kind of data. So we can do all of that fairly quickly. Within months, we can identify individuals, map them to pedigrees, identify these pedigrees. Where things kind of slow down is when we have to get that biological sample to sequence someone. That's a challenge. There's unfortunately no existing biorepository here at the university where we can just say, okay, we've identified these individuals, give us their DNA or give us even better their sequencing data. That doesn't exist, at least it's sort of siloed. So we were fortunate to be able to contact some of these individuals. This is one particular pedigree. All of the folks that are shaded are folks that we performed the whole genome sequencing on. We did some analyses with the help of Mark Yandel, and nothing really shook out. And that's kind of a challenge with these families, because as you can imagine, this person is probably related to these folks over here, who are all cases by our definition, and they share very little, relatively very little of the genome. So it makes the analysis complicated, hard to really pick out genes and variants that might be of interest. However, we were fortunate that we were able to hone in on this range that had a number of individuals that met our phenotype of rapid decline. And this individual is this individual here, and this individual here. And we were able to expand that pedigree quite a bit. And we actually found out that this person who we didn't have any data on in terms of being in the EMR system had end-stage renal disease. So when we expanded that family and sequenced them, considering this person as where the variant could be coming in, we identified this variant in a gene called FRAS1. It's this arginine to cysteine variant. And we performed that analysis with Mark Yandel's help. At the top of our, in terms of statistics, FRAS1 emerged as our leading candidate, which is incredibly exciting for us. When we identify variants like this, one of the first things we do are go to these large databases, how common is this variant? And that database that we typically use is called NOMAD, represents the number of different populations that you can see that this variant was seen very, very rarely, only in 5 individuals of European ancestry, 1 of African ancestry, out of over 100, almost 150,000 individuals. Yet we saw it multiple times in this family. So that was exciting to us. We went to the literature, and there's some biology behind this gene. It's associated with the glomerular membrane and maintaining the integrity of that membrane. Those of you who are clinicians know the glomerulus is the functional unit of the kidney. So here we have this link of this gene that emerged from our analysis. Of course, when we find things like this, we like to dig deeper. We leverage some publicly available single-cell sequencing data, and we saw that in individuals with diabetic kidney disease versus control, we see reduced expression of this gene, both in the mesenchyme and in the podocytes. That's quantified here with the diabetic folks in purple. We also see that this gene has reduced expression in the tubular cells in individuals with diabetic nephropathy relative to those who don't. Looking at the literature, we're very excited to see that just a couple of years prior to us identifying this gene, this group from Finland published a genome-wide association study in type 1 diabetics, looking at genes that might cause diabetic nephropathy. They looked at 2 signals. One signal on chromosome 6 at the HLA region, probably not a diabetic nephropathy locus, locus for type 1 diabetes. They had the second signal here at chromosome 4 that met their level of genome-wide significance. If you zoom in on that, this locus zoom plot, you can see that variant is right above the FRAS1 gene. So incredibly exciting. As geneticists, we always like to have this replication of our findings. This had been put out there, although this gene wasn't particularly excited about that hit. Now we see this in a family. So is this variant functional? That was our next question. We have this statistical support, which is great, but we didn't want to just push away from the table. We wanted to take it the next step forward, see if it has some sort of function. So my lab was not really great at this, but we're really fortunate at that time to bring on Kiwa Chen, who was previously with Kevin Whitehead, who I believe was in this department, maybe still is.
Yeah.
Okay, so he was a former technician with Dr. Whitehead. We're fortunate to bring him along. And he leveraged CRISPR and HEC293 cell and introduced this variant and then ran a couple of assays to see whether or not it could impact the function of those cells. He did 2 assays. One was a scratch assay or wound healing assay, and the other was a cell migration or cell adhesion assay. And in addition to introducing our variant, this arginine cysteine in these cell lines, we also introduced a synonymous variant just to ensure that the CRISPR wasn't mediating it in the effect that we saw in these cell lines. And then we also hit these cells with heron and knocked out FRAS1 completely and looked at that effect. And he grew these, so he did that wound healing assay, grew these cells in high glucose, and I think it's going to appreciate that relative to variants that don't have the or cells that don't have mutation. those cells migrate much slower in that high glucose environment. And he quantified that on the right-hand side, of the screen. Below that image is his quantification of these cell adhesion adhesion assay. Interestingly, when he repeated this same experiment in the presence of normal glucose, he did not see a difference between the synonymous variant or our gene distinct variant, supporting our hypothesis that this is probably a variant that is mediated by being in that high glucose or diabetic milieu. And that is also what we see in the pedigree. You do have some carriers on this variant, but they don't have diabetes, they don't have that diabetic or that disease phenotype. So we're really excited about this. We're trying to, and we're working on this publication, or at least this manuscript, and hopefully submitting it soon.
So are you able to produce this in a more physiological system? Like, you can't.
So we haven't taken any further than this
Okay.
in terms of other cell models or even
Sure.
animal models. We've kind of left it here and
Right.
are hoping that we can package that.
Yeah, sure.
Some of the work that we've been doing most recently is, and I'll talk about that next, is where a lot of our efforts have gone. So we think that this is an exciting approach
No, I don't.
where we're leveraging families and identifying variants, but it's also challenging. And we were fortunate to identify this family prior to COVID. Interacting with families has been a little bit more challenging since. And so we've kind of put that on the back burner in a sense, but we do have all these families that we could certainly continue this approach with. So now I'm going to change gears and tell you about some work that I've been doing with my key colleagues, Will Holland and Scott Summers, and it's really been an exciting area for us in terms of scientific collaboration, friendships that we've made with them, and really the direction that it's helped us to take the lab in. So with Scott and Will, we decided to do some targeted sequencing at the time because, again, whole genome sequencing was too expensive. We could do targeted sequencing of Scott and Will's favorite genes. Those folks who know Scott and Will could have guessed that those genes come from the ceramide or sphingolipid pathway. And we sequenced a lot of the folks that we were recruiting from dialysis centers and nephrology clinics. And we came across these three individuals who, when we sequenced this targeted panel of about 30 genes, had a variant in a gene called ASA2, which I'll mention briefly. But these are the pedigrees of the three folks that we recruited. So the individuals on the proband, highlighted by the arrow, and these are the family history that they told us. Huge families, lots of obesity, lots of diabetes. We don't have much additional phenotypic information, but all three carriers had end-stage renal disease, and two of them had cardiovascular disease. We assumed that when we did this, so one of the first things that we do is we go to this database called Nomad and ask the question, how frequently is this variant seen? Wasn't seen at all in Nomad. Hundreds of thousands of people wasn't seen at all, yet we found it in 3 people. We figured, okay, these folks must be related. We can do some use some genetic tools to try to get at that. Turns out they weren't related. So we looked at their chart a little bit deeper and we found, so here's their clinical characteristics again, all diabetic, all have eGFRs less than 15. 2 out of the 3 were on dialysis. But all 3 of them came from either Tonga or Samoa. That was exciting for us, particularly being a New Englander, not knowing that that was a community that was really vibrant here in Utah and Salt Lake City. So kind of eye-opening for us. We sequenced a few more folks, found 6 more carriers again, all from Tonga, Samoa. Most of them had type 2 diabetes. Most of them had obesity. Most of them had very advanced, all of them had very advanced kidney disease and eGFRs less than 15. So that was really exciting for us and eye-opening. This variant was not seen, again, as I mentioned, in this Nomad database at all across any of these frequencies of these populations. It's a valine to leucine change that's highly conserved, predicted to be pathogenic by various computational tools that we can leverage to try to get a sense of is that variant important or not. Interestingly, it's also located at the very first nucleotide of exon 5. So this is intron 4, this is on the negative strand. Intron 4, exon 5, intron 5 would be down here. The very first nucleotide, and if we use some computational tools, it is predicted to impact splicing. So I'll talk more about that in just a minute. So we knew that this variant wasn't present at all in this Nomad database, but native Hawaiian and Pacific Islanders aren't among those populations that were listed there. So we reached out to our friends across the country and asked them to look at this variant in their various cohorts. One of them being the Simone adiposity study, which had whole genome sequencing of about 1,300 people. They saw the variant at about the same frequency we did in our cohort, so about 5%. We asked our friend at USC to look at the multi-ethnic cohort, which has various ethnic groups, included about 1,000 Hawaiians, and they saw that in 4 individuals. So a much lower frequency than we saw. We suspect those people are public admixed. We looking all of us Native Hawaiians to Congress at the time it was 150 people, not seen at all, and not seen at all in the Maori population of New Zealand. And that kind of makes sense, at least with our hypothesis that this variant was probably introduced to one of these 2 islands, Samoa or Tonga, some time ago, several thousand years ago, perhaps, and rose to a higher frequency in those islands because each of these islands are populated isolates. So variants that are very rare elsewhere can rise to what we call a common frequency. And geographically, it also makes sense that we're not seeing in Hawaii, it's about 3,100 miles away, New Zealand about 1,500 miles away. So it sort of makes, we haven't investigated that much further, but there's some really interesting population genetics going on here. But we do think that it originates from these islands. And we'll probably be digging into that a bit more as we grow our cohorts. So going back to the variant itself, ASA2, those who are familiar with this pathway, and I won't speak too much about it because there's an expert in the room who might say that I just spoke, but this is the ceramide de novo synthesis pathway, where this enzyme ASA2 comes into play. It's a ceramidase, breaks down ceramide right here. So these are the ceramide which we have to bodily lipotoxic molecules that build up in our cell and lead to all sorts of deleterious effects. This enzyme functions here to break down ceramides to its byproducts, smithine and S1 phosphate. So one of the first things we asked, and this is with Will's help, do these carriers of this variant have increased ceramides? And it turns out that they do. So we had 5 carriers that had the variant, 5 non-carriers who were also cystic governors, had diabetic disease, and 10 non-carriers. You can see that those folks in the blue here, our carriers have increased C16 ceramides, the really bad guys in the ceramide literature, and they have decreased S1 phosphate, that byproduct, and increased ceramide to S1 phosphate ratios. So this is from serum medical patients, which is really exciting for us. I won't go into all the work that Becca has done. Becca is a really talented graduate student in the Summers Holland lab who has really taken on functionally characterizing this variant and helping us to understand whether or not it is biologically important. One of the first things she did was to introduce this variant into HEC293 T cells using CRISPR, and she noted that this variant led to a much smaller product. It's a wild type variant, a product with her RT-PCR. She sequenced that. Turns out that it leads to skipping of exon 5 in frame, the 177 base pair amino acids. So 59 nucleotides are just gone from that protein. It's in brain, so we believe that protein is being made. And she showed that in this assay. Becca has gone on to express this mutant or deleted protein in cell lines and show that it lacks catalytic activity, which is really exciting. So the business end of this protein is this 59 amino acid domain. And she's shown that in a construct that lacks that, it does not degrade ceramides. So that's really exciting. What's happening schematically is this. So in people who carry the wild type allele, they make a protein that has all exons of the gene, including the catalytic domain. Carriers of this variant have this complete skipping of exon 5, have a protein that lacks exon 5 and ceramidase activity. So it's really cool and continuing to characterize that and work out exactly what that's doing. So identifying that variant and work that we had done prior with Will really kind of came to a head when we looked at some of our prior work in a family that we identified that was actually a family from Maine that had a very similar phenotype to the patients that carried this ASA2 variant. All were diabetic, all had very advanced renal disease. These folks had low adiponectin levels. They had this mutation and adiponectin of the dominant negatives that we worked out with the help of Joe and Chris Mione, and it had increased ceramide levels. And this goes back to work that Will published earlier in his career that showed that adiponectin, through its receptors, it actually functions as a ceramide. But for us, this is really great. We had these two genes that we identified in these two different groups. One was a family, one was a population that has this variant that all converge on the same pathway in the same aspect of this ceramide pathway. So as we would expect or hope to see patients who have impairment of either of these two proteins, we would see increased ceramides, decreased sphingosine, as we showed in that paper. In the data that I just showed with you, we do see that in either carrier of our adiponectin deletion, which I won't go into much more, or MESA2 variant. So I'm going to skip over to this just to kind of move on to our next section. So this was really, again, eye-opening. So being a guy from New England, coming to Utah and seeing this really high rate of disease in our community. And we saw this empirically when our study coordinators go to the dialysis centers. About a third of those patients were Native Hawaiian Pacific Islanders. 30% of Utah's population is not Native Hawaiian Pacific Islanders, about 1 or 2 or close to 2%. So this really increased burden of disease that we saw with our own eye. We know about 50% of the NHPF population is overweight, 40% obese. About a third of those will go on to develop diabetes. That's 3 times the rate that we see in other populations, particularly of white individuals. And half of those folks will go on to advanced kidney disease, and yet we don't really know the causes of that. As a geneticist, we know that folks that are Native Hawaiian Pacific are not being included in the studies that we're doing. So they're not benefiting from all these great breakthroughs that we're seeing with the advances in science. This is just a summary of the GWASs that have been done to date. 80% of those studies have been done in European individuals. We see other groups that are being represented in those studies. This sliver right here is actually an aggregation of folks in Oceania and the Middle East. It really represents about 0.1% of all genetic studies. Huge disparity when we think about the burden of disease relative to their inclusion in studies. And what's even, I think, harder to understand is that we've known that there's been this genetic link for over 40 years. And this was a study by Paul Zimmet, an epidemiologist from New Zealand, who put out this notion of a diabetes genotype in Native Hawaiians and Pacific Islanders. And he surveyed a bunch of folks and kind of posed this hypothesis, yet not a whole lot of ground has been made up in this population to better understand their disease. So we think there's an opportunity here to really do something that's impactful for our community. Folks may know that Utah has the 5th highest Native Hawaiian Pacific Islander community. So we think that it's really important work that we can do. But there's some challenges to this. And I've highlighted sort of 3 challenges that my lab thinks about a lot. So there are really relatively few existing cohorts. 2 that I mentioned are the Simone Adiposity study, the multi-ethnic cohort, about 7,000 patients total. That might sound like a lot, but when you're talking about genetic studies, that's relatively very few. So to try to mitigate that or make advances in that area, we've established what we call the Utah Pacific Island Diabetes Study. Started that about 1.5 years ago. We're approaching 1,000 patients that we've enrolled right here in Utah. And we're also leveraging other data sets, including things like MVP, which is a study of 1 million veterans, and it includes 0.7% Native Hawaiian Pacific Islanders. It may sound small, that's 7,000 people, but they're really powerful data set. Something which I'm going to try to get into, man, we have to breeze over quickly, is that Native Hawaiian and Pacific Islanders aren't represented in the reference genome that we use to analyze our data. That may not mean a whole lot to a lot of folks in this room, but you can kind of think of it as a massive puzzle, and you're trying to put those puzzle pieces together. To do so, you need to have the puzzle box as a reference. And when we sequence people, we get all these little pieces, and we line it up to a reference genome. This linear genome is a string of A's, C's, G's, and T's, and that genome is derived from a person of European ancestry. And that works pretty well, but every once in a while you'll get that piece that has some variation that's specific to your population that isn't European, and you throw it away because you can't align it. That piece could be important. So we think that's a critical deficit in the field. That's a thing called reference bias. So I have a student who's taken on this challenge to devise the first specific ancestry reference pangenome, and that's referenced here. It's not just a genome of one person, but many people, so you can align all those species. I won't talk about this much. I'd like to talk a little bit about the reference genome, but we also know that there is not a good existing genotype imputation panel. Again, it's not represented in that, so we can't impute variants that are specific to that population. And that's what's kind of shown here. When we use existing populations or reference panels in the red, we can impute okay, but as we add individuals, 25, 50, 100, 200 individuals that are of our target ancestry, we get improved imputation. That's what's shown by those incremental lines there. So these are two resources, actually three, that we are developing to really advance this research. Due to time, I'm not going to go into what the different sequencing technology, short read versus long read sequencing. Both are really exciting. We are, for the work that we're doing now, we've been doing some long read sequencing where we can do de novo genome assembly. So we don't need that puzzle box, that map. We can put that together on our own. And that's by using long reads, assembling those, and doing this assembly without having that puzzle box. And that's what's shown here, but I don't have, just for sake of time, I'll kind of brush over some of these things. So we challenged our graduate student, Connor Littlefield, to do this, but he assembled a Pacific ancestry pangenome, and he leveraged 23 samples from our collection from individuals whose origin was from across the Pacific. So individuals from Tonga, Samoa, Fiji, the Marshall Islands, Pohnpei, Guam, in the Philippines. And we identified these folks by their reported grandparents. Where did they come from? So all four came from these various islands. And he performed this long read sequencing, very large sequencing assemblies, 16 kb that it then assembled to create this pangenome. And that's what's represented here. So I didn't go over it much. They haplotype pangenome research consortium, which includes 47 individuals from various ancestry. This is a Chinese pangenome. This is the pangenome that Connor assembled, which is in a bioarchive preprint. Our data is performing very similarly, if not better than a lot of those other cohorts. So for this type of work, you don't need thousands of folks. This is 47 individuals. This is 58, ours is 23. And you can take that and assemble these genomes. Really quickly, this is, these are our 23 samples color coded here. These are individuals that are included in the haplotype pangenome consortium, these 47 individuals. And when we look at the cumulative growth, what are we gaining when we add additional pangenomes or individuals to this pangenome? We see that our pangenome colored here, we're gaining about 31 megabases of additional data. So every time you add more diverse groups and look at these pangenomes, you're increasing your coverage across the genome. You're identifying a new sequence, could be disease related, could be related to other things. In total, we identified 34 or 3.4 million new variants that would otherwise not be discovered using existing resources, and almost 5,000 structural variants. So this is great, right? We have this resource that we identified. It includes specific element ancestry. Does it work in terms of improving variant calling in our population? So I always tell my lab that this is fun stuff, but our goal isn't to just create these resources. We want to identify variants that predispose to disease. So does it work? So to test that, and Connor's still working this out, what he did was he took 200 individuals that were specific on ancestry that we had short read data on and mapped it to this pangenome that we developed. Do we see improvement or gain in terms of variant calling? And the answer is yes. So if you take those 200 sequences and align it to the haplotype pangenome research consortium, which does not include NHPIs, you identify about 68,000 variants from chromosome 21. When you combine our pangenome with that one, you identify over 2,000 more variants. So we are discovering potentially novel variants for sure. that could be related to diet diseases that we're studying, diabetes, kidney disease, etc. If we expand that across the genome, we expect to identify approximately 120,000 novel variants. So now our next step is, can we identify variants that are relevant to the phenotypes that we're studying? And very quickly, my last slide. This is not only great for structural variants, but in Connor's PacBio long-read sequencing, he's able to identify large deletions or structural variants that would otherwise be missed by these short-read technologies. And one that we're particularly excited about is this 258 base pair in-frame deletion in a gene called lubricin that is associated with type 2 diabetes and gout. And gout is a prevalent disease in this population. He's currently working on genotyping this variant in our 200 samples, but right now he's seen it in 3 of our 23 folks who made up our pan genome. So there's about 10 minutes left, and I do want to leave time for questions. But really quickly, I want to thank all of our collaborators with SPAN departments and divisions for all of their help. Our various funding agencies, including the Center for Genome Medicine, the Martin Lowell Foundation, but most importantly, our study participants. We certainly couldn't do this work without them. We also have a number of community partners who have been strong advocates and supporters of our work, giving us great advice on how we can do things, and not just do things, but do things right. So we really appreciate their expertise. And I will close with this, sort of our team in action. And I'm happy to take any questions.
Yeah, this is really quite exciting.